Hello everyone, so we're going to talk about the scientific method, and if you look at this graphic organizer, it will kind of help you remember the steps. So as you could see, we first start with making an observations, and from our observations we start asking questions, and which we research and then see what other scientists have done, what we know already, and start formulating a question. And this question should be a testable question. And from this question, then we form a hypothesis, a statement that we can test. And then design our experiment and see whether our data will allow us to accept our hypothesis or reject it. And we'll further discuss the details on how you would have a good experimental design that includes the independent variable and dependent variable and constant, and of course the control. So let's start with the first step, making observations. So here's a picture of a frog. What do you observe? And if you see that it's a frog, doesn't a frog have 4 legs? So these frogs were observed in Minnesota near ponds, and a lot of people were alarmed that there were not just 1 or 2 frogs with deformities, but a lot of deformities. And the big question would be, what is causing these deformities? Is it something that can affect people? So after you made these observations, you want to identify and define the problem you want to investigate. It must be in the form of a question. So what is causing the frog deformities? Now, first you want to do some research, right? Gather the information related to the problem. Read, observe, measure, do some field studies, take samples, take surveys. What other studies have done on the same topic? How do frogs develop? What percentage of frogs have these deformities in a particular location? Number of other species in the pond with deformities, types of pollution, change in the amount of ultraviolet rays, sunlight, exposure on eggs, pond environment. Do other animals exhibit these deformities, or what can cause developmental problems? So after you have done research, you want to formulate a hypothesis. And a hypothesis is an educated guess which has a possible solution, prediction based on an idea which will be tested by a particular method, and this is referred to as a research-based hypothesis. It must be a testable and a falsifiable statement. It's based on research and previous experience. And part of the hypothesis is formed by a statement: if I change the IV, then the DV, by using a certain technique, and a stating the method and idea being tested. Once we have formulated a hypothesis, we're ready to start designing our experiment, right? So we design an experiment to test the hypothesis, and one of the most important things in an experiment are having these 2 groups. An experimental group, which we're going to change the variable, and a control group, what we're going to compare it. So in our experiment, the experimental group is our test group, in which the conditions are kept the same except for one thing, that is our independent variable. And the second is our control group, which is what we're going to compare it to, and we're not going to change the IV. We're going to keep it what's in the normal situation. Variables in this experiment: an experiment should include an independent variable, and you want to only change one thing. And a good way to remember what is independent variable: I for independent, I change. And the independent variable is, when you plot it, it's always on the x-axis. The dependent variable, or the DV, is the variable that changes because or responds to the IV, or is what you're going to measure. And the dependent variable is plotted on the y-axis. And then you have the constants, which are factors that you keep the same. Now, collecting and analyzing data. Data can either be quantitative or qualitative, and organized in tables and graph to help scientists visualize trends. So if you notice, here's a graph. One of the important components of a graph, you've got a labeled graph with the units. You have a title, and then here you have a chart. Again, units are very important. Now, there are two types of data, qualitative versus quantitative. So we define qualitative data as something that is observed but not measured. So things like colors, textures, smells, taste, its appearance. It's qualitative, quality. Quantitative is something that deals with numbers, something that you measure, like length, height, area, volume, weight. So it's quantitative. So here's a cup of latte. What are some things that are qualitative and quantitative data that you can collect on this cup of latte? Now, looking at qualitative versus quantitative, a latte has a robust aroma, frothy appearance, strong taste, as in the burgundy cup. Those are qualitative data. What can you measure about a latte? How much, the volume, the temperature, how much it cost. Conclusion. Now, this is the meat of your experiment, where you take your data, analyze it, and see whether your data supports it or rejects it. This is where you decide whether your hypothesis was correct or incorrect. And a conclusion must have these three parts. You state the hypothesis, state whether it was rejected or accepted. Were there any sources of error? Follow-up experiments, future studies, just kind of summarizing your results. Another important thing about experiments is repeated trials. The more trials, the more certain you're going to be about your data. So repeat, repeat, repeat. And then when you're ready, you would publish or communicate your findings. So scientists will have their experiments peer-reviewed and verified, and then they would share their work by publishing their findings in professional journals. All right, and some of these questions that scientists would ask about a scientific work is, did they follow the correct method? What was the control? Did they measure what they're saying they measured? Does their data really show a trend, or what was their statistical analysis? How certain is the scientist about their conclusion? What could there be a future study? What could there be an experimental error, and so forth? So this is again steps that generally all scientists would follow, and again we use this to find solutions and using evidence to support scientific knowledge.